this is where I am now in the Hebrew University. And we will talk today about modeling using SAXS data in the context of integrative structure modeling. So what is integrative structure modeling? So in principle, we want to use information which can be of any type. Today, we mostly focus on SAXS data, but it can be obviously structures which we will use. It can be structures coming from AlphaFold or ZetaFold or new accurate modeling programs. And we would like to use all this information to come up with a final model or models that satisfy the data. And so what we are doing is developing computational methods that are able to integrate all this information. And so what are the modeling tasks? Obviously protein folding, which can be homology or comparative modeling or ab initio modeling, assembly of multi-domain proteins, protein dynamics, so it's more of flexible assembly, assembly of multi-protein complexes. So these are the tasks, and we will talk about each one of them today, and you will see examples from the tasks here. So let's start with the basics. Any task like that requires a tool for calculating the SAXS profile from a model. We are talking here about atomistic models. We view it as a scoring function in the context of integrative modeling. And this is why when I started working on SAXS many years ago, the first thing we did with Michal is developing FoXSDock, which is a calculator for a theoretical SAXS profile. So what it does, it takes a model, it converts it into a theoretical profile, and then it fits it to the experimental profile. And so this calculation is performed using the Debye formula. And the scoring function is chi or chi-square. We can get rid of the square root here. And our goal is usually to minimize chi and to bring it ideally to 1. And so the scoring function takes into account excluded volume and iteration layer density. So we need to take this into account. And because of those two things, we have two parameters. So one of the parameters of FoXSDock is C1 that takes into account the excluded volume of the protein. And this is basically increasing or decreasing the atomic radii by 5% variance to account for the excluded volume. And the second parameter, C2, accounts for the iteration layer density. So remember, we have two fitting parameters, and we will get back to them later. We gathered a data set of several structures and experimental data sets, and they all fit well, and we published the FoXSDock, and it's been in use for around 10 years already. But that's all great, but the reality is slightly different. So when you measure your data set and then you come and fit your structure, quite often you will see that they don't fit at all. So here you can see many examples that are very, very far from a perfect fit. And we can see it using the profile displayed on log scale, but we can also see it using the residuals, which is basically what we see in the chi-square formula. It's a experimental profile minus the computed divided by the error. So, so you can clearly see that those don't fit. And there are several reasons why your calculated profile doesn't fit the data. So first of all, data quality. I'm not talking about it. I think it was yesterday in the workshop, but you have to make sure that your data is great. The next reason is missing residues, sugars, or other atoms. And we are going to talk about it. It's important. The other two is heterogeneity, compositional and conformational, or it can be both. And so I'm going to talk about all those soon. So let's start with missing residues. So I am going to change what I share now. So remember that a SAXS profile is basically your distance distribution. And if you have missing residues, then you are missing parts of distance distribution, but you are missing much more. So if you have 5% missing, you are going to miss around 10% from the distance distribution, which is a lot. So if you have a short protein with, let's say, missing a few missing residues that can be a HISTAG, you can get quite significant misfit. So now I will share my screen. So this is, no, this is the wrong one, sorry. This is the one I want. So this is the web server. And now I'm going to input a short protein. So it has a PDB code, which the web server will fetch from PDB. And I will add a SAXS profile here. Okay, so the profile is added and I just click submit and then it is calculated. So it will take a few seconds. Here you go. So here we can see the result of the calculation. The black is the experimental profile and the red is the calculated one, and this is the residual. And this is the protein that we used for calculation. And below we can see the numbers. So this is the PDB, the chi-square is 9. And here we have the two fitting parameters, C1, C2, and the number of atoms are the subduration. But first I have a question to you. So what do you think? Is it a good fit to data or not? So you can use maybe the thumbs up, thumb down, or whatever we have in Zoom, and let me know what you think. Okay. What else? You don't like it? Yes, you're right. This is not a great fit. So maybe it looks fine here, but if we zoom in, I don't have a mouse, so it's difficult to zoom in, but trust me, it doesn't fit anywhere. And so, so based on the residual, we can clearly see that it doesn't fit. This should be ideally straight line. And we have a huge misfit here. And so what happens is that this protein doesn't have a HISTAG. So let's add the HISTAG. So how can you add the HISTAG? Anyone has ideas?
Using AlphaFold?
Yes, that's probably the default today.
Yeah, because it's so automatized, you can really grab it.
Yes, it's so easy to use AlphaFold to add the HISTAG. But there are many other options. So I like using Modeler for this purpose. So whatever you like. So this is a Modeler in Chimera, Chimera before Chimera X. So you can see that it uses Modeler, and here you can see 5 different models with HISTAGs going in all the directions. Here you can see this HISTAG that was missing, and you can see that the fit improves a lot. Okay, and we can do the same on the web server. We can upload those 5 models, and we can see that everything improves. So let's do it. Okay, so here is the server. And now I want to go back. And I have those models zipped already. So I upload them here. And the SAXS profile. Okay, so we submitted.
I just want to mention that the zip file have the both files, PDBs, the histag and non-histag.
Maybe, I don't know. Maybe only histag.
We'll see.
I'll see, yeah. Okay, yeah, it has both. Good. So the yellow one is the original one. The color is random, so I'm sorry, it doesn't look very well, but you can see that the red fits much better. So this is, you can see here the residual is now almost a straight line, and you can see the fit is very good. Chi-square is 1.2, which is almost perfect. And what we can also see that previously Fox was overfitting. So you can see this is chi-square, it was 9, C1 was 103, and C2 was 3.4. 3.4 is the duration layer, which means things were missing, the histag was missing. So it tried to compensate for the missing histag by having a larger iteration layer. That's why this parameter was high. But now it's within reasonable range, so we know that we fit well and we don't overfit the data, which is very important. Okay, so you can also, you can use FoX on the web server. You can use it in Chimera, and I guess we should ask Chimera X to edit. You can also use it as part of IMP package. Just install it. And if you go to the website of IMP, they also have notebooks. So if you like Colab notebooks, they have notebooks with Fox as an example. So it's very easy to use in the Colab as well. Okay, so I hope now you understand how important it is to add everything. Now here you can see an example of missing sugars. So all the atoms are here, but this is a protein with lots of glycans, and it doesn't fit very well. Once we sort of grow these glycans longer, we get quite a good fit. So you have to add all the histags. If you have glycosylation or other modifications that have significant volume, they also have to be present. And the last addition, which is very recent, and we are working on it with Michael right now, we are adding magnesium ions for RNA molecules. So RNA binds magnesium ions that are required for proper folding and charge neutralization. And obviously missing ion density can result in incomplete distance distribution. So same story. And here you can see RNA doesn't have all the ions, and you can see that it doesn't fit the data very well. So the question is, can we predict the positions of magnesium ions given our structural model of RNA? And because this is very recent, we decided to address this question using deep learning models. So what my student Dan did, he used all the RNA PDB structures to extract all the magnesium ions that are close to RNA. And basically we trained the network using around 100,000 examples of magnesium ions and the RNA around them, using water molecules as negative examples, because the network to learn to distinguish between water and magnesium. And so we tried two networks, convolutional neural network and graph neural network, and eventually graph neural network was much better for this task. And the network given a probe or position in 3D space next to RNA predicts if it can be a magnesium. So this is RNA. We surrounded with different probes, and we select those that are likely to be magnesium. So to add the probes that are predicted by the network to run RNA, we can use a SAXS profile. So we start by adding single ions, and we check how well it fits the SAXS data. This is chi-square score, and this is the number of magnesium ions that we added. And you can see that the score improves up till it reaches some optimal number of ions, and then it sort of goes up again. So we can use the SAXS profile to select the ion positions that were predicted by the network and to further optimize their positions. So this was about adding all the missing atoms. And now I will talk about heterogeneity. So many times, even after we fill in all the missing parts, we still don't fit the data. So you can see here, this is 3 structures. Those are crystal structures, and you can see that all the tails were added, but none of them fits the data within the noise. And already based on those 3 structures, we can see that what is the problem. So anyone wants to guess what's going on? Do they look similar to you, the structures?
The disordered regions are different.
Disordered, yeah, but it's quite a big structure, and the disordered region is not that big relative to the whole structure. There are several domains here. I try to look at the distances between the domains in the 3 structures.
Okay. Just the motion of the N-terminal domain.
Yeah, so we can see that the distance here is shortest between those domains, N-terminal and C-terminal. And here it's probably the largest, here it's something intermediate. So based on those structures, we can say that this is the flexible protein, domains are moving. Okay, so what do we do when we have something like that? What we need is we need to sample the different structures. And the sampling is dependent on the situation. So we can have something that is almost completely rigid and not moving, and we can have slight domain motion, and we can have a large scale domain motion. So domain can move quite a lot, and we can have completely disordered regions. And this is quite common. So if you look at the structures, and today, actually, if you browse AlphaFold database, you can see it quite well. Previously, you needed specific tools to predict disordered, but now it's all over those predictions. We know that around 40% of structures have short disordered fragments, and over approximately 10% have longer disordered structures, and this is PDB. But actually, SwissProt and obviously AlphaFold, you can see disorder everywhere. And so to fit the data properly, we need to model this disorder. So the good news is that SAXS data is capturing this disorder, and we can now try and model this. But the bad news is that data interpretation is challenging. Okay, so we need to figure out what to do. Okay, so what do? We work as following. So we have a heterogeneous sample. We know that we have several conformations of the same protein or complex, or even we can have a compositional heterogeneity, which is monomer or dimer or different assemblies. And to fit the data that is heterogeneous, we need a multi-state model. So what is a multi-state model? It's several structural states and usually other parameters. And for SAXS, the other parameters are just the weights. So, so what do? How do we model? How do we generate multi-state models? So our input is the structure. It has to be an atomistic structure. It can be a model, and now it can easily be AlphaFold model. And it has to be obviously SAXS profile, and then we decide on a list of flexible residues. So if we want to sample a motion between the domains, we don't have to sample everything. We can limit the sampling to the linkers between the domains and those disordered regions. And what we have to do is to produce this list of flexible residues. And then we can use a sampling algorithm to generate many conformations for our system. Then we can use the FoX to calculate all the profiles. And then we need to select subsets of conformations that fit the data best. And those subsets are actually the multi-state models. Now we have to be very careful not to overfit the data with too many conformations. So analysis is very important. We have to understand what are the features that are responsible for those multiple states and what do we feed the data with. So how can we sample the protein? Here we use the motion planning approach that is coming from robotics. And this is because proteins have those angular degrees of freedom similar to robots. So this is a rotational angle, and obviously phi and psi are rotational angles. So those approaches transfer to proteins very well, and we can rely on what was developed there. And we explore the space that is accessible to the protein without collisions. So we don't want any steric clashes between the domains. So we are looking for the space of all possible conformations without steric clashes. This is not necessarily lowest energy minima or anything like that, but this can be done very, very fast. So if we want to sample interdomain motion using molecular dynamics that will go to the minima, it is really time consuming. Here we can sample quite a lot within minutes or maybe a couple of hours and generate quite a lot of conformations that cover the space very well. So next we select best scoring multi-state models. We start from models of size 1 and we go to models of size 2. We cannot keep all of them because it's quite a lot. So if we started with, let's say, 10,000 models of size 1, then size 2, it's 10,000 squared. So it's quite a lot. So we will only keep, let's say, 10,000 best scoring ones based on chi-square fit to data. And we will extend them to multi-state models of size 3. Again, keep the best scoring ones and so on. So this approach is called branch and bound, extend and then keep the best scoring ones. And we continue that until chi-square can no longer be improved. Now we can improve our score to account for multiple states. So this is chi-square when we have a weighted average of the profiles for all states. And the weight here, W, can be optimized using non-negatively square fitting. So this optimization is analytical, and we can solve for weights very rapidly. And the big advantage is that we basically have a single set of fitting parameters for the whole multi-state model. So it will be a single C1, C2 pair for a multi-state model. If there are 5 states, it is still 2 parameters and not 10 parameters, which is very important if we want to avoid data overfitting. Okay, so if we go back to
could I ask a quick question?
Sure.
So I was just curious how you decide what sort of the most parsimonious model is in a way, because one could imagine that the most parsimonious one is the one that has the fewest conformations on the one hand, or on the other hand, one might imagine the most parsimonious is the one that's sort of uniformly distributed among all possible conformations.
So
how do you think about that?
This is an excellent question. I'll get to it in a few slides. Okay, so just hang on 2-3 minutes and I will respond to that. So let's go back to this example first. And so we said this is domain motion, and indeed, if we apply this algorithm, we can get an excellent fit using 2 conformations. So you can see it really opens up much more in solution, and 25% of open structures and 75% of closed structures explain the data perfectly. The chi-square is basically 1. Now, let's go to an extreme example. So this example is a Prion protein, and this is 50% disordered. So we know that the monomer is basically disordered, and it then only has the structured fragment of 3 helices. It's also not very tightly packed. And so those are NMR structures. And so to get SAXS data, we had to use obviously FPLC SAXS technology, otherwise it aggregates. And here you can see the difference. Red is monomer and blue is the one that aggregates without FPLC. And so now let's see how we can model this. And I'm showing this recording of the web server. So this is a MultiFoXS web server. And it gets, as I said, the PDB of the input protein. It gets a list of flexible residues and the SAXS profile. You put your email address, and after a while, you get a link to the results. So here it is. This is the modeled linker. You see it's completely disordered. And this is a list of flexible residues. Basically, this whole linker was given as flexible residues. And we submit, and we get an email. And this is how the results page looks like. Here are a few plots. So the first plot is score as a function of number of states. So we know that if we add more states, the score, chi-square score should go down. And indeed, we can see it here. It goes down. Next, we have the fit between experimental data in black and different states, multi-state models. And here is a plot that has some analysis of radius of duration of the results. So let's start with single state models. So we can see that the best scoring 1-state model can fit the data quite well. The chi is 1.16, which is not bad at all. So this data set basically can be fitted with a single state despite being a disordered structure. But if we go to two-state model, it improves very, very slightly. So now you will see how it looks. So if it's a two-state model, it comes next. This is the best scoring two-state model. And here you can see that the chi is slightly better. And you can also see the two states. And what you can see is that one of them has a RG of 31 and the other one has a RG of 40. So this is much more open. The linker is more open compared to this one. And if we go down, we see three-state models, four-state models, and so on. And I think now we can go back to your question. Okay, before the question, I want to advertise BilboMD. I think, Michali, will you talk more about it? So BilboMD can do something very similar to MultiFoXS. The main difference is that it uses high temperature MD for sampling of the linkers. So for some systems, maybe this robotics-based sampling is better. For some systems, maybe this one is better. But you can try both and see what works best for your system. And here there is a very nice interface to define rigid bodies and flexible linkers.
Just a comment. We'll not talk too much about the BilboMD. It's just the same calculator what we're using in the MultiFoXS, and multi-state modeling is exactly identical in BilboMD. It's just a conformational sampling. It's done differently.
So the sampling is different, and it can depend on your system. So yeah, so you can try both, as I said, both. Ideally, they produce similar results. Okay, and now back to the question. So the question is, okay, if we use a small number of states, then we really, we are not overfitting the data, but it's not clear if we represent the heterogeneous sample. And it was very obvious for there's this example that I showed you before of Prion Protein, which is 50% disordered. It's quite clear that it has more than one conformation in solution. On the other hand, if we sort of keep adding states, we are in the danger of data overfitting. And so how do we balance those two? So what we do is the following. We compute multi-state models for a low number of states. We don't go very far, usually up to 5. We can go up to 10, but normally it's up to 5. And we also set a lower bound on the weight of each state. So normally it's a parameter, but the default is 5%. So a state has to contribute significantly to improvement in chi-square to be included. So this sort of takes care of data overfitting, few states, and a significant weight to the state. But on the other hand, we don't want to look at a single multi-state model. So remember, our process of generation of multi-state models usually goes by at least 1,000. So we can start with 1,000 single-state models, and we generate 1,000 two-state models and 1,000 three-state models, and so on. So if we look at the end-state models, we will usually have 1,000 of them. So we will analyze those 1,000 or at least 100 best scoring ones, and we will look for conserved features among those models. And those models are obviously different because it's different conformations, but we can look what is most common. And the easiest feature to look at is probably radius of gyration, because it sort of represents how compact or open the structure is. So I don't have the example. Okay. So we analyze and we can always see that, for example, if we have two states, then one can be always open and the other one closed. They can be different and they do represent the variety of what's going on in solution. So we get, let's say we get 1,000 two-state models and they represent the heterogeneity of the sample. So we will never say that the two states that are best scoring according to our score are the two states. But if we take 100 or 1,000 our two-state models that we produced, this is already becoming representative of what is going on in solution. So I hope I answered your question. Okay.
Yeah, if I could just add one more thing. It seems that there's going to be some manifold of possible models that fit the data equally well.
Exactly.
And then you have to decide some additional criteria on top of that as to which ensembles are more likely than others.
No, I think whatever fits the data equally well, this is what represents the heterogeneity, because we know that those are flexible systems and they obviously sample lots of states. So all those combinations are probably valid, and without additional criteria or without, let's say, orthogonal data set, we cannot exclude any of them. If we have additional data, let's say cryo-EM or cross-linking, then we can start maybe excluding, but without additional data, this is what we have. And I think this represents quite well what is going on in solution for those flexible systems.
Okay, thanks.
Okay, so let's continue. So let's say that we have our data set. We used great scoring functions. We used great sampling algorithms, and we modeled something. And so now we ask a basic question: is the data is satisfied? If it's satisfied, then it can be a single structure or multiple structures. And if it's multiple structures, then, as I said, more data is needed if we want to sort of exclude some of the options. But this is obviously the maximum that we can get from SAXS data. But if the data is not satisfied, then we need to think about sample heterogeneity and flexibility. So first, let's try to fit the structure rigid with all the missing parts. If it doesn't work, we go to heterogeneity and we try flexible parts, or maybe it's compositional heterogeneity, monomer, dimer, or other assemblies. So this should be the order in which you fit the data. Okay, questions till this point? And I'll show several examples of the same principle with other data sets. This is EM data 2D, this is antibody, and we can sample the antibody, same sampling approaches, and then we can fit it to 2D class averages. So this is very low resolution data, but because of the Y shape of the antibody, we can clearly see what's going on, and we have atomic resolution structure. So we can find the 2D, we can find 3D conformations with 2D projections. So those are conformations, and this is a 2D projection, and you can see that this projection fits the 2D class average quite well, and those are other conformations, other projections generated from multiple conformations for this protein. This is a similar principle from residue dipolar coupling data. So those are two ubiquitin domains. They're moving with respect to each other. They're covalently connected. And each single domain fits the data perfectly, but together the structure doesn't fit the data well. And again, if we use a multi-state model, here it's a three-state model of closed, partially open, and completely open, we can get a perfect fit to the data. And cross-linking, same story. So cross-links, we can see blue color are satisfied ones, and the red color are the ones that are not satisfied. Usually we use 35 angstrom cutoff or not satisfied. And here, this is again two-domain protein. So domains move with respect to each other. And using three, so you can see that there is no single structure that fits the data, lots of red unsatisfied crosslinks here. But together, those three structures satisfy the data very well. And you can see here in this histogram of distances that there is only one crosslink that is within 39 angstrom, but the rest is satisfied. So when we have three structures, we only require that one of them satisfies the crosslink, and it can be unsatisfied by others. And again, same idea, but different data. Okay, so now I'm going to complexes. And we can model protein-protein complexes using SAXS data. It's harder than Legos because there are no instructions. So we are going to use the SAXS as an instruction to help us. So 2D problem is very easy, but somehow in 3D, it becomes much more difficult, and we don't know how to fit those pieces together. Even if you 3D print them and play with them, it's very hard to put them together when we go to 3 dimensions. So what we are looking for, if we fix one protein, let's say we fix the blue protein, and then we are looking for the transformation T, which is 3 rotational degrees of freedom and 3 translational degrees of freedom. And those 6 parameters tell us how to bring this protein to the blue one so that they are in contact with each other. So we have 6 degrees of freedom. Additional problem is that proteins are flexible, even if they're rigid. Side chains can move or loops can move. So this is not domain motions, it's just the rigid domains, but this is really small scale motions, but they still complicate the docking because they change the surfaces, and the shape complementarity is not perfect anymore when we use the structures or models that were not determined together, right? And so what happened is that methods that sample complexes, we call them docking methods, they can generate thousands of models, and usually they will include a model that is close to correct. So sampling is not a problem, but the problem is identifying those correct models. So this is really finding a needle in a haystack, because normally less than 1% of models are correct. It's like 0.5% or even less, and the rest is incorrect models. And but we know that additional information helps a lot, so a SAXS profile should help us to find the needles in a list of many models. And so that's where we developed an approach called FoXSDock. So what we do, we generate multiple docking conformations with a tool called PatchDock, and we use Fox to sort of calculate theoretical profiles for all those docking conformations. And then we fit the experimental profile and compute the score. So again, the bi and chi-square, same story. So many different models, and we can sort them by the score. FoXSDock is also available as a web server if you want to use. You need to give it the 2 proteins in PDB format and the SAXS profile. And so, again, same approach can work with different data sets. So SAXS, it can be EM2D, as I showed you for antibodies, 3D, and cross-linking. And we tested it on those 5 data types. So this is shape information, SAXS and EM. And the crosslinks give us medium range distances. We also had data set that can give us information about the interface. And what was interesting is that, so what we did, we simulated large amounts of data for a huge benchmark of almost 200 test cases for docking. And we could see which data is most informative for those complexes. So what we found is that if we look at the standard docking and we look at the top scoring solution and top 10 solution, because beyond that it's becoming hard to look at those solutions. So just docking without any data sets, only 10% of the cases we have the correct solution at top scoring, and it's 24 for top 10. And we don't have confidence estimates, so we don't even know if it's correct or not. If we use the SAXS data, we double our success rate, and with the EM data, we actually triple it. Cross-linking also doubles the success rate. And 80% in top 10 is sort of the upper bound. For some reason, it's very hard to go beyond that. Now, the interesting question was which combinations of 2 of data sets can help us. So obviously SAXS, EM, and EM were not complementary because they are a sort of shape information, but we could get a huge improvement when we combined shape data sets with cross-linking data sets. And this combination brought us the correct model to be top scoring in 50% of the cases. So SAXS goes very well with crosslinks. Now, so basically, if you can get a single data set, try EM probably. 2 data sets, you can get SAXS and crosslinks. Both are very fast experiments. And what was interesting is that back like 2 years ago and 4 years ago, CASP was actually running data-assisted experiments. I don't know why didn't they do it this time. And there was an interesting complex that had the SAXS profile and cross-linking data sets. So, which means that we had a very high probability of success here, at least 50% for top scoring, and we could submit 5 models. So it had 7 interprotein crosslinks and no structure. The problem was that there were no structures for the 2 proteins because CASP is actually a folding competition. So we were given sequences and we had to fold and dog the 2. So we decided to see if any of the predictors had good models, and because basically CASP makes the models available once web servers produce them. And we selected models that satisfy the intraprotein crosslinks because intraprotein crosslinks were also available. So here are some proteins with okay, no crosslinks here. So for the first chain, there were 7 crosslinks, but no convergence. There were like 20 models that were completely different. The second chain did converge on a good model. So we decided to dog 20 models, best scoring models for this chain with this model for the second chain. And once we docked them and started to select based on interprotein crosslinks and SAXS, we could see convergence. So we basically were obtaining clusters of very similar models. So here basically docking helped the folding, and we could select the correct model, and it indeed turned out to be very close to the crystal structure here on the right. So this is a nice example of integration of two data sets, SAXS crosslinks, and obviously shape complementarity and statistical potential for interface scores. And now I will use the remaining time to talk about antibodies. So antibodies are very interesting proteins because for many reasons. So we produce them as a result of exposure to pathogens or vaccination, but antibodies are also a huge class of therapeutic molecules. And one of the main challenges is determining where the antibody will bind or what is the epitope. And SAXS profile can be very useful in this context. We, in collaboration with Arvind from ADIMAP, we collected the SAXS profiles for EGFR, the blue protein here, antibody and their complex, and we did it for 5 antibodies with known structures because we wanted to see how well the method performs. And here you can see the SAXS profiles, and what do you think? Does it fit well? No. Yes. So based on those lines, right, we don't like it here. We can clearly say it doesn't fit. The chi is also very high. So they don't fit the data very well. And this is crystal structures. So what is going on? So what is happening? Let's start with EGFR. EGFR turned out to be slightly flexible. This domain can move. Those are 3 crystal structures, but it doesn't move a lot. And actually the main problem was the glycosylation. So it has 10 glycosylated positions. And if we grow those structures a little bit and combine them of the glycans, we can get quite a good fit to data with one state model. Now the problem is actually antibodies. So here we only use the FABs. And you can see that if we take the X-ray structures, it doesn't fit completely. This is 5 different FABs, and the chi-square is very high. Now what we can do is that we can move the domains of the FAB relative to each other like that. It's called changing the elbow angle of the FAB. And this improves quite significantly. So here you can see the chi-square becomes better, but it is still very far from what it should be. So what else is there? So we noticed that several crystal structures in PDB have what is known as crystal contact, where antibodies stack with respect to each other. So here we can see it's called head-to-head stacking, where the loops of the antibody touch each other. There are even domain swaps of the antibodies, where they swap the domains of each other. So there is some evidence to interactions of antibodies. And so we gathered some of them from the PDB and tried to see if we get better fit if we use those dimers. And surprisingly, or not surprisingly, the fit improves quite a lot if we include a really small percentage of those dimers. So here you can see the percentage of dimers. It's really 2 to 5 more or less. And the chi-square is much better when we include those dimers compared to without dimers. So probably some of them do dimerize in solution. And now we can use docking to see if we can generate those complexes. So we have SAXS profiles. We used the SAXS profiles of the Fab and the antigen to obtain models for antibodies and for the antigen, and it can be single state or multi-state model. And now we dock all the conformations or all the models to each other, and we obtain many complexes, and now we can continue as FoXSDock. So we check using Fox the fit to SAXS profile. We use the statistical potential and the new deep learning model to check, to test the fit at the interface, how well the interfaces fit together. And we combine those scores and produce top scoring models. And here you can see an example. So what I show you here is called funnels, scoring function funnels. On the x-axis, we have interface RMSD, how far we are from the correct model. And on the y-axis, we have the score. So we want our score to go down and have funnel shaped as the RMSD goes down. And here indeed, we see that with SAXS, all the correct models, and I show them here in red. So every dot corresponds to a single docking model. The red models are the ones with RMSD below 4 angstrom. They all have a SAXS chi-square score that is low, and there is nothing above some threshold, which means we can exclude all the models above this line, which is quite a lot. And this is really the power of SAXS. So it allows us to exclude many, many models. And now we can look at the statistical potential score, and we can see that it doesn't separate the models very well. And the good models have scores all over. Similarly, the contact net score, also interface score based on deep learning, it excludes quite a lot of models which are given a score of 0. It excludes quite a lot correct models, but we can see here there is one good model that gets a good score. And once we combine all the scores, we can see that the SAXS and contact net together, they sort of rank it as top scoring ones. And if we look at the results for 5 complexes, we can see that using SAXS chi-square, 2 of them do rank the correct model as top scoring, but 3 don't. Using SOP, statistical potential, only 1 of them ranks it as good scoring one. Contact net ranks 2 as top scoring ones, but the combined scoring does much better, and we can see that 4 out of 5 actually do rank the correct model among top 5. So integration of information really helps us a lot. And I will finish with nanobodies. So nanobodies are basically single domain antibodies. So if this is antibody with light and heavy chain, nanobodies only have heavy chains, and if we only use this variable domain of the heavy chain, it has the same properties because this is responsible for binding, so it can be used as a binder. And when the pandemic started, we were interested to see if we can develop nanobodies to neutralize SARS-CoV-2 virus. And we were working with the lab of Yishi in Mount Sinai now. And what they do is that they have a technology based on mass spectrometry to identify all the antigen-specific nanobodies from the serum of the llama. So the approach works as follows. You immunize the animal, And after a couple of months, you get the serum, and you identify the sequences that bind to your antigen using mass spec. And now they express around 100 of them, although they can identify thousands of sequences. And what we want to do is, again, find the epitopes. So same problem. So because they have mass specs, they didn't get SAXS profiles, but they got crosslinks. And we use the same approach with crosslinks to rank different complexes based on how well they fit the cross-linking data. So here you can see in gray, white, the receptor binding domain of SARS-CoV-2. And here are the different nanobodies. And you can see how docking models place them all around different epitopes based on different crosslinks. Okay, so I will wrap up here, and I hope that you could see that you can do quite a lot of things with the SAXS profile and the integration of different data sets. So those are the tools that we use, and probably more. And I guess you understand that using different types of data, we can optimize the accuracy, precision, and resolution of structural models, and we can construct single state and multi-state models for the large and dynamic complexes. And hopefully we can also infer something about the function from these models. So I want to thank to everyone who was involved in those projects, especially Michal, who helped and participated and inspired all the development of those programs. And we still work on this. I guess I want to give you one more link. So if you want to use docking with nanobodies and SAXS, I have a notebook. I'll just put a link in Zoom here so that you can go over and use. So this notebook has basically an example of docking with the SAXS profile, and you can always open it, change it to your proteins, your PDBs, your data, and try it. And it has all the different programs inside, so it brings it for you and you can run it. Thanks.